I'll move to my talk about real-world data. Well, we do not discuss real-world data or real-world evidence most of the time. I thought as an introduction to highlight what is the difference between real-world data, real-world evidence. Actually, there is a small subtle differences. These are data relating to patient health status and or the delivery of healthcare routinely collected from a variety of sources. So real-world data is information for routine practice. Real-world evidence is the clinical evidence about the usage and potential benefits or risk of a medical product derived from the analysis of the real-world data. And you generate real-world evidence if, for instance, you want to use it for drug approvals or at least to complement this. So I think, and I'll try to convince you, and this is now established since many years, that real-world data or evidence is clinically relevant for physicians and payers. They can allow for hypothesis generation. They can allow for comparative effectiveness, treatment strategy assessments, quality of care, but also effectiveness. And it's not here about opposing prospective studies versus real-world data. Actually, they go hand in hand, especially that sometimes not everything can be assessed through a dedicated prospective study. So when it comes to regulatory decision making, real-world data after the approval of a given drug are extremely important to first analyze the landscape, because you may get an approval of a drug in third line, and then you would notice that in the real world, actually everybody's using it earlier. It is, of course, for post-approval drug safety assessment, the famous PASS studies, which are usually mandated by regulatory bodies. It can allow also to detect later drug interactions, medication errors, but also it can support registration and label expansion because the drug is becoming more familiar and more widely used. And this is very relevant for rare diseases. And of course, the real-world data evidence can allow for economic modeling to inform pricing and forecast. And this is, of course, important for the regulatory bodies, but also payers. In regulatory decision-making, assuming you would like to use real-world data, real-world evidence, obviously, drug dosing is important because you may have an approved dosage, but if you have a decent, significant amount of data using another dosage, actually, it can allow to assess alternative dosing regimens for an established medication. Also, it's about identifying target drug combinations for testing in future trials. And I mentioned this, label expansion, new indications, new populations, additional endpoints. And we've seen this in many hematology drugs, which were approved with a given relatively stringent label. But we know today that thanks to real-world evidence and a few other trials, they are more widely used. Okay, what type of real-world data or evidence can we collect? Well, actually, we can collect demographic, social determinants, medical information, pharmacy information, genomic information, laboratory diagnostic, radiology, imaging, radiation, oncology, patient-reported outcomes. All of this can be collected, and I think it will become more and more easier with all the AI tools to collect and extract this information, which can be merged or aggregated. And also, interestingly, it's not a fixed time point. You can collect these data over time. So how to select the data source for real-world data? Well, I think most of you are familiar with the electronic health records. whenever applicable, if it is available in your country, medical claims, the registries, and of course in Europe we are all familiar with the EBMT registry, but also in North America the CIBMTR, but also it can be patient-generated health data. And of course, for the sake of time, I will not go through the pluses and minuses for each source, but I think all of them have strengths, but the limitations are what they are. And at the end of the day, irrespective of the source, one must acknowledge that the challenge with real-world data is usually about the quality, because obviously this is not monitored like what you see in prospective trial. So you can see some heterogeneity and some variable quality. Study designs. Well, of course, you can design a prospective observational study to collect real-world data, and this is a very high level of quality. One can use retrospective observational study. It could be a descriptive study, but we can also have actually pragmatic clinical trials and externally controlled trials with real-world data being used as control in a given prospective trial. So the weakness is I insisted about the quality, and this is, for instance, about the missing data because they are not available, they are not collected, they are not accessible, and sometimes you can miss some key data because you are relying on routine practice. So one cannot easily guess what were the inclusion and exclusion criteria that were used by the brain of the physician to decide on this treatment or this treatment. And of course, in general, when we treat in routine practice, well, we don't have a predefined study endpoint. And of course, there are many other confounding factors. Completeness of available variables can be a matter of concern, lack of randomization, statistical plan, written protocol, and as I mentioned, no monitoring and little or no control over the quality of the data collection. Despite this, I think more and more the community is very keen on generating these real-world data and provide real-world evidence because the field is moving very quickly. We're doing registration trials more and more, at least in hematology, with a limited number of patients, and this is why after approval, you need to have a clearer picture about the drug, and this is where real-world data can be useful. And here I'll show you an example how we can, through real-world data, actually reproduce and feel more comfortable about the use of a given drug. It was mentioned by Dr. Wolf earlier that belumosudil has been approved for the treatment of steroid refractory chronic GvHD. It's a ROCK2 inhibitor, and this is a phase 2 multi-center study that evaluated 2 dosage to at least a way of administering belumosudil in patients with chronic GvHD who had received 2 to 5 prior lines of therapy. This was published by Dr. Cutler, and you can see we are talking about a response, overall response rate, which was the primary endpoint, the best overall response rate around between 74 and 77%. So these are the other endpoints, the median time to next treatment, the safety profile, which was excellent, a little bit of fatigue, diarrhea, nausea, but they were always very mild. And actually, these results were further consolidated with the long-term follow-up, actually, where we had an update about these results with 3-year follow-up. So registration trial, very reassuring with more than 3 years of follow-up. So how does it compare? How these data compare to the real world? And we were very fortunate to have very quickly a French compassionate use program of belumosudil. And we were able last year to collect the data, and that was a very careful, meticulous collection of information of 68 patients. You can see majority had moderate, severe chronic GvHD. The median time to chronic GvHD was 6 months, up to 50. And you can appreciate the incredible number of treatment lines before getting the compassionate belumosudil. The median treatment lines before belumosudil was 3, going from 2 to 8. And you can appreciate the details here. Ruxolitinib, ECP, outside ruxolitinib, all the rest are not approved in Europe. And this is our actually selection of empirical use of some of these drugs historically in chronic GvHD. Well, you can see here the results. The response over time to belumosudil at 3, 6 months, but also the best response. And this is absolutely, when you consider CR, almost 15% CR, 43% PR, and we had 37% who had a stable disease. And this is a median follow-up of almost 11 months. And actually, the median time to best response was around 6 months, and the median duration of treatment actually was between 8 and 9 months. So you can see here, just looking into the response rate, we are exactly very comparable to the registration trial, which is very reassuring, especially that this population, in my opinion, is more advanced, more heavily pretreated than the registration trial. Overall survival, 6 months, 90%, 12 months, 85%, and the famous failure-free survival endpoint, which also is a great 80% at 12 months. You can see the causes of death and the different adverse events, again, in line with the what has been published in the registration trial. So here, I would like to share with you a typical patient case study which we encountered, and this is a young lady, 49-year-old, AML, first CR, match unrelated peripheral blood stem cells, female to female, 10 out of 10, reduced intensity conditioning, flu-bu-ATG, standard of care, calcineurin inhibitor plus methotrexate for GvHD prophylaxis. She engrafted day 16, and by the 4th month, we could see some more mild oral lichenate lesions and dry eyes. Mild chronic GvHD, managed with topical steroids, but also continuation of tacrolimus. Unfortunately, she experienced progressive skin thickening and new dyspnea and worsening dry eyes. So you can see maybe things are not moving in the right direction. At the 10th month, you can see the evaluation of chronic GvHD in the skin, in the lungs, in the mouth, in the liver, and unfortunately, this is severe chronic GvHD. You can appreciate the skin involvement, the FEV decline from 85% to 58% predicted with the typical images on the CT scan. But also you have a liver involvement, which is quite interesting. And of course, the treatment started standard of care, and again, it was discussed nicely by Dr. Wolf, prednisone 1 milligram per kg per day, and continuing tacrolimus. Well, despite 8 weeks of done systemic corticosteroids, the skin sclerosis and lung function continued to worsen, and the liver function continued to be altered. So, in theory, if you have followed the presentation of Dr. Wolf, this patient has a steroid refractory chronic GvHD because she progressed despite receiving more than half a milligram of per kilo per day of prednisone. So what would you do next? Well, next step management to be discussed, ruxolitinib, considering ECP, or actually moving to a ROC inhibitor like belumosudil. Obviously, if we follow the label in Europe, I think the straightforward answer would be ruxolitinib. This is what we have achieved thanks to the REACH3 trial. Considering ECP is a bit more tricky, and I know some colleagues may combine ECP with ruxolitinib, but again, we don't have a sort of a validation or standard of care for this. This has been published in a smaller series from Germany. It's definitely feasible. ECP is safe, of course, but you can see these questions. And of course, Dr. Wolf also mentioned that ROC inhibitor like belumosudil is already approved in China from the second line. So if you have this possibility, you may also consider belumosudil in this patient. And of course, you may have also the issue of those patients who, for whatever reason, they are not very frequent, but cannot receive or cannot tolerate or whatever contraindication to ruxolitinib. So in summary, I hope you are convinced that real-world data and real-world evidence can provide unique insights into patient journeys, treatment strategies, and outcomes. They do complement pivotal clinical trials because they can allow to assess effectiveness, safety, quality of care in a more diverse patient population. We do acknowledge all the limitations, lack of randomization, heterogeneity, missing data, but they are highly valuable for hypothesis generation, comparative effectiveness, and practice optimization. In the specific field of chronic GvHD, given the heterogeneity of the disease, also given the rarity, because at the end of the day, we're not speaking about diabetes or heart problems. So the number of patients is relatively limited. So we'll not be able to randomize hundreds and hundreds of patients, and depending on some specific localizations, we may wish to use real-world data to further assess and investigate innovative therapies. And this is exactly what we did with when analyzing the French compassionate program of belumosudil, where you could reproduce and validate the prospective data to guide best clinical practice. And of course, registries are extremely important. I didn't mention biobanks, but of course, something extremely important in our field, especially if we want one day to develop biomarkers and to have predictive factors for chronic GvHD and for outcome. Obviously, these biobanks can be important, and you will hear later about the quality of life and the PROs, but again, these can be generated. And I'm convinced personally more and more that thanks to all the AI tools, this will become more and more easy to generate very good real-world data and evidence.